Hello, this is lab number 8. My name is Roberto Blanco. It's the Biochemical Test Lab, part 2, and my lab partner is Eros Aranda. Um, okay, Introduction. This lab explores the lysine decarboxylase test and the orthine decarboxylase test. Basically, this to identify gram-negative bacteria if it's gonna be metabolizing, basically. It's gonna tell us some colors. It's gonna be a differential test, a selective test too. So it's gonna tell us like, hey, I wanna let grow this bacteria, and if it does, it's gonna change the color. These biochemical tests determine if an organism produces specific enzymes capable of breaking down the amino acids lysine or orthonine. Okay, again, it's just gonna be specify if it's gonna break it. The results based on color change in the media, Leah, and orthorobot help differentiate between organisms like E. coli, Margomela morgini, P. mirabilis, and S. tritophorum. Successfully interpretation of this reaction is key to identification of these test organisms, including gram-negative unknown. Materials and methods. The materials in this case, it was just Leah and orthidine broth, basically test tubes. Cultures, we use C. fruthley, E. cloquet, E. coli, M. morgani, P. mirabilis, P. aeruginosa, and S. tipinmurium. Will be the procedure. Basically, we use the lines in the chromato test, stop the bottom, and stick the slide and record the results. And okay, let it grow for a couple days. Whoa, 1 day. Orthine decarboxylate test. We inoculate the respective broths with the started organism and record the results. So basically, the procedure for the lice first test, we basically grab from our plate, from our broth, grab some, put it inside the tube, inoculate it, a streak, and that's it. And another one, it was basically again get some sample from a tube and literally shake with our loop, put it in, let it grow. Okay, so results. In this case, E. coli show metabolism because it changed color to purple. So it actually grows. For P. mirabilis, it does reaction, but because if it's yellow, it means it's negative. The S. tryptophan is actually reacting because it changed to purple. And Morgan also too, but you can barely see some growth. That's the only weird thing. And the unknown 271, actually, you can see a change of color to purple. It means it's positive, and you can see growth. So the only one is negative is gonna be P. mirabilis in this case. Actually, it was very interesting. I actually like this one. I just like the color change of every single one to identify if it's gram negative or gram positive. The results for the orthine decarboxylase. E. coli changed color to purple, meaning it was a reaction. Cifrodin, this one actually changed color like orange, purple, but like very, very like transparent. So maybe it was a problem on the inoculation on this case because it might be gram negative, but I'm not too sure. It might be that would be the case. I hope it wasn't. So let's say that this one didn't react. Our unknown 21 actually reacts, it changed color fully to purple. And conclusion. Actually like this lab. It was pretty interesting. It was fun. Super simple, nothing crazy. We actually did it super fast that I recall. So this laboratory investigation successfully employed the lysine decarboxylase test and the orthine decarboxylase test to evaluate the specific metabolic capabilities of several gram-negative bacteria, to observe color changes in the media, the key indicators of positive or negative decarboxylase activity, allow for the differential between the test organism, including E. coli, M. organs, and the rest, right? The ability or inability to decarbonyl lysine or provides vital information for the systemic identification of these organs. Basically, it's saying that it's gonna show the activation of catabolism. Therefore, the lab achieves its objective by generating data that can be interpreted to distinguish the test gram-negative species, completing a crucial step in their identification process. I mean, we actually see which organisms were gram-negative. the ones that were purple, basically. Uh, that's it. Thank you so much.